Hi, thanks for coming to the presentation of Chelation Partners. My name is James Drage. I'm managing director of the company and was the original seed investor. I invested in Chelation Partners because it has a new way of looking at treating infectious disease. And I did that because I have a very personal story. My daughter, Jordan, when she was 17 years old, caught an antibiotic-resistant infection and almost died. Thankfully, she's still with us today, and after several different antibiotics, she got better. But I learned through that experience from doctors and research that the way we treat infection today needs to be improved. The standard of care could be better. And so with Chelation Partners, I think we found a better way to treat infection. And to explain how, I'll turn it over to Michael Weckert. Off to you.
Thank you, James. I appreciate the introduction. And just to let you know a little bit more about myself, I've been working in development of pharmaceuticals for over 28 years. And the area that I've worked the most is infectious disease. Infectious disease is an area where arguably the greatest accomplishments in human health have been made in the last 100 years. And that's because of a combination of improved hygiene, especially water treatment, the development of antimicrobial agents, and the development of vaccines. But as the SARS-CoV-2 pandemic has shown us, we're just one new infectious organism away from a repeat of our history of 100 years ago with the Spanish flu pandemic. Not only are these kind of emerging infectious organisms a threat, but we have a slow-moving pandemic as well that's going on all around us. And that's the threat of the fact that our pathogen drugs that we currently use are losing their potency, like James was describing with his daughter. Pathogen-resistant, antibiotic-resistant organisms and pathogen resistance to other organisms is widespread. And if it's not unchecked, we expect more deaths from drug-resistant infections than from cancer by 2050. There's a second part of this threat that's also disturbing. The reverse economics that currently doom new antibiotics that are being developed. And that's because they're reserved for last-line use. Only after everything else fails do we use new antibiotics for fear of development of resistance. And as a result, companies are going out of business. With low sales due to the reservation of those products, they can't be a going concern. We've had 3 major antibiotic companies in the last year go bankrupt. As a result, large pharma is also fleeing the field and saying it's just not worth it because we can't recover our drug and development investment. So we desperately need a new approach or we're going to have millions of people die from previously treatable infections. And that's where Chelation Partners comes in. We're doing something dramatically different. Instead of focusing on the pathogens, which has been our method of preference for the last 4 or 5 decades, we're actually working on defending the host. Pathogen attack relies on vulnerabilities that the pathogen has, but those vulnerabilities are overcome when the pathogen mutates. And that's the development of drug resistance. When you help the host defend itself, it's a game changer because you can now improve a broad spectrum of host defense against pathogens. What we're using to do this is Dibby, an ultra-potent, non-toxic iron-binding polymer. It combines a generally regarded as safe polymer used pharmaceutically since the 50s with the iron-binding domain of a drug, deferoprone, that was approved in 2011. As a result, this combination is non-toxic. It's demonstrated in 14-day, very high doses administered, which is different than all prior chelator drugs, which have significant toxicities and black box warnings. Dibby is actually able to bind 3 different iron molecules with each molecule of Dibby, trapping them with extraordinarily tight binding. Now, why are we focused on iron and iron binding? Well, because iron is really important in infections. So iron is required for human life. A lot of our enzymes have iron as a critical cofactor. And we actually manage iron levels very carefully to maintain health. Parasites also require iron, and you know where they get it from. They get it from us. And as a result, whenever we're infected with a pathogen, there's a tug of war going on biologically. The pathogen is trying to obtain iron, and the body's trying to withhold it. It comes along, and it actually scavenges the available iron, tipping the balance in favor of the host by removing that essential biomaterial from availability to the pathogen. As a result, this starvation of that biometal causes the pathogen to slow or stop growing and either dies on its own or becomes very vulnerable to the effects of other commonly used antimicrobial agents like antibiotics. But that's not all. The ability to collect iron by Dibby is actually really potentially valuable in a number of other hypermetabolic conditions like sepsis, where the cytokines are being overproduced by the immune system, or something like cancer, where a tumor is rapidly growing and needs a lot more iron in order to continue to grow and metastasize. We don't have time for those stories today, but Dibby overall is not only a broad spectrum antipathogen, but it also has applications in sepsis, cancer, inflammation, and a number of other disorders. Now, Dibby satisfies what I would consider to be the ideal infectious disease product profile. You've got an agent that has single micromolar minimal inhibitory concentrations against many strains of bacteria and fungi. It's potent against drug-resistant pathogens. And there's no way for pathogens to gain resistance to this approach because there's no genes on the pathogen that can be mutated to avoid the need for iron to live. Antipathogen synergy has already been demonstrated in a number of published models using multiple drugs in multiple different animals. As a result of this, we don't have to be concerned about reserving this for last line use. And that changes completely the economics of development and use of this compound. Dibby has single micromolar concentrations that are effective against gram-positive, gram-negative, and fungal organisms as shown in the table on the right. In animal models, Dibby alone as a monotherapy reduces the bioburden of infectious diseases by many orders of magnitude. This applies to gram-positive organisms, gram-negative organisms, and also drug-resistant organisms, both in superficial infections like wound and nares, but also in pulmonary infections and systemic infections. In a second animal model shown on the right, the combination of subtherapeutic levels of Dibby and an antibiotic known as fusidic acid is more effective in reducing the bioburden of infection than the actual clinical concentration of fusidic acid that would normally be prescribed. That's showing the synergy of combining this with antibiotic approaches as well. But what we really care about in the moment is COVID-19. And there are a variety of applications for Dibby in response to COVID-19 infections. We have potential for direct antiviral effects, and we have potential for secondary clinical benefits, some of which I've already alluded to. And those include the ability of Dibby to synergize with other antibiotics, to treat drug-resistant secondary bacterial infections, particularly in patients with pneumonia, which is quite common, as you know, with COVID-19 patients. The second secondary clinical benefit is the suppression of cytokine storm, which is a major contributor to mortality in COVID-19 infections. On the direct antiviral effects side, there's evidence pointing to a significant role of iron and iron chelation in COVID-19 infections. So we know that iron is required for viral replication, particularly for RNA viruses. But we've also recently seen in a published analysis of over almost 1,600 COVID-19 patients that the most prevalent comorbidities associated with severe cases were conditions characterized by iron overload, too much iron in the plasma or intracellular spaces, allowing the pathogens to grow and proliferate. In other diseases and other viral infections, chelators have shown significant antiviral activity. And this applies to other RNA viruses that are like SARS-CoV-2. Several independent research groups have speculated that iron chelation therapy may be very effective for COVID-19. What's been missing is until now, nobody had a non-toxic iron chelator like Dibby to use. We actually have two collaborations underway right now testing Dibby against SARS-CoV-2, one at UCSF and one at McMaster, and we should have the results within weeks. Dibby's direct antiviral effects also include the potential to reduce viral attachment and uptake because it turns out that a portion of the SARS-CoV-2 spike protein has structural homology with hepcidin. Hepcidin is the regulatory hormone in humans that regulates iron uptake and export, and it binds to a receptor called ferroportin. Well, it turns out that Dibby strongly downregulates this expression of ferroportin, as you can see in the chart at the bottom. If it does this in SARS-CoV-2 infections, it would significantly reduce potential binding and uptake sites for that virus, slowing the infection. In addition, it may act like a natural flavonoid to reduce viral activity. Flavonoids have already been shown to have activity against other coronaviruses. It's been speculated that their activity is due to their iron chelating ability. Dibby has not only structural similarity to these flavonoids, but obviously has very potent iron chelation activity, far more potent than that seen in these natural flavonoids. Dibby is ready for GLP toxin and IND filing. We have done toxicology studies by 4 different routes of administration in 3 different species at very high doses and have yet to observe any significant adverse events. We have production at 100-gram scale up underway, and a simple 3-step manufacturing process that leads us to be potentially ready for clinical trials within a few months. We have an accelerated plan for developing Dibby in COVID-19 and preparing it for clinical testing such that we could have conditional approval for emergency use within a year. This depends, of course, on the ability of regulatory authorities to authorize the use of this in COVID-19 patients rather than going through the normal pathway of normal volunteers. But either way, we should have this in people for testing as soon as possible. And as you know, the circumstances are pretty unusual. Dibby, though, is a pipeline in a product. By that, I mean it has an extraordinary diversity of medical applications and thus an extraordinary business opportunity. We have the immediate COVID-19 response that we've pivoted to from the development of our core business, which would be a sustainable anti-infective business, where we meet the ideal product profile for a new anti-infective agent. But we also have potential upsides in sepsis and cancer and inflammatory disease and others. And we're protecting and pursuing this by patents covering our active pharmaceutical ingredient and its use in a variety of infectious diseases and other diseases like cancer and sepsis. Because the drug can be administered many different ways in many different dosage forms and strengths for many different disease states, it has the potential for multiple out-licensing opportunities, as well as making it a potential attractive target for M&A and IPO in the future. Important to note, though, is that our initial focus here on COVID-19 is not essential for the long-term company success. Right now, what we require to move this forward is $9 million that will allow us to complete the pre-IND work and the initial maximum tolerated dose study, preferentially in COVID patients if approved, otherwise in normal volunteers. We actually have an opportunity to leverage some of that investment because the first 2.2 million of this investment unlocks 2.5 million of additional capital through several Canadian grant and stimulus programs. These include an ACOA grant that the company has already been approved for $450,000 and a $3 million Canadian matching investment that would accompany any equal investment. So investors need only provide 6.5 million of that 9 million that we need. And I just want to leave you with 2 thoughts. The first is that Dibby is a genuine game changer in infectious disease. It really meets the ideal product profile. It's hard to think of anything that would have a better profile in order to be useful for not only our initial emergency that we're all struggling with now, but also future pandemics and future general antibiotic use. Secondly, it's extraordinary that a drug like this would have secondary potential in so many other major illnesses like cancer and inflammatory conditions. Thank you for listening. One last thing, our deck will be available attached to our bio profile. But if you wish to hear more, please don't hesitate to schedule a meeting or reach out to us. Thank you. Bye-bye.